Hello, good morning, everybody, or good evening, depending on where you are. Uh, First of all, I want to thank the organizers of this symposium for organizing this nice conference, as well as the sponsors, Recent Biosciences, for giving me an opportunity to present some of my work. So it's a pleasure, and I'm really looking forward to presenting some of my results briefly. I just want to say that this is a pre-recorded talk, so if you have any questions, I will be around the question and answer session and the discussion session, as well as a recent biosciences breakout session, which will happen later today, I believe. Today being the 29th. Yes, so with that, I will get started. I think perhaps many of you might be familiar with the fact that proteins are essentially the targets of most drugs. And even the most common drugs or most studied drugs can actually have a large number of protein targets which are unknown, which means that their mechanism of action remains unknown as well. And so if you can identify some of these targets, it can help us not only elucidate molecular mechanisms through basic and fundamental biomedical research, but it has a lot of benefits for the clinic as well, because we could potentially design better drugs or repurpose existing drugs for different clinical diseases. And to give a very good illustration of why this is important is from this paper, somewhat recent paper, which speculates that actually a lot of the drugs, cancer drugs that are in clinical trials are having their primary effect through unknown mechanism of action. So, I mean, it's not really, it's kind of scary when you think about the fact that, some of these some of these drugs that are in clinical trial are having their effects through unknown mechanisms. So we're really just gambling by doing these clinical trials if we're not even sure, which proteins or how exactly these drugs are working in vivo. And that is actually not so surprising when you think of the fact, how new drugs are commonly designed. Typically, you have your protein or small number of proteins of interest, and you screen a large number of drugs against those proteins, depending on your assay. Of course, this will also take account into the high complexity of the environment in which these proteins exist in the cell. And what I mean by that is we know that many proteins engage in protein-protein interactions. We know they're modified in several different ways, and as well as we know that many of these proteins are in different compartments within the cell. So a lot of these things can affect how a protein interacts with a certain drug or inhibitor, for example. And so there's a large number of methods which try to figure out some of these targets, but typically they're limited by the fact that you can only perform some of these methods on a small number of drugs. So an ideal method, you would screen a large number of proteins against a large number of drugs for activity. And so this is what I try to address with the method and workflow I'm going to present here. So this is actually based on thermal proteome profiling. And the advantages of this approach that I find is that it's really nice to scale up to a plate-based format. And of course, I find that if I couple this to a label-free proteomics approach, it can be a very robust way to perform some of these target engagement assays. Just a brief background on thermal proteome profiling or cellular thermal shift assay for those who might not be so familiar. The basic concept is that if a protein is interacting with a drug or inhibitor, that will shift the melting point of the protein. And what I mean by melting point is the temperature at which 50% of the protein is still soluble. So if you can imagine a scenario where a protein interacts with a drug, that changes the temperature at which the protein melts, or at which the protein is being stabilized by interacting with the drug. And this you can read out using mass spec based approaches by basically measuring the soluble proteins over the course of different temperatures and determining the melting point and seeing if it is shifted in as a result of treatment with a drug. And the advantages of this approach is that you can identify not only interactors of potentially different drugs, but also a lot of indirect interactors. And what I mean by that is protein-protein interactions, for example. However, some of the limitations of this approach is you need to measure the melting point. And some ways to get around that is basically, instead of measuring the melting point, you basically integrate the melting peak. And what I mean by that is you can imagine a scenario where a protein is being stabilized by a certain drug. And instead of trying to measure the melting point, you basically pool soluble fractions from different temperatures into one fraction. And then as a readout, you just measure the protein abundance of that sample. And so this is an approach that I'm utilizing, or a variation of it. And of course, I'm not the first one to use this. There's a couple of different groups that have reported this technique, including the group from Sweden here, our Zubrab group. So just to test this method with the label-free quantitative proteomics approach, I treated HepG2 liver cell line lysates with either DMSO control or staurosporin. And as you can see from this volcano chart, I plotted the median intensities, log2 intensities of the proteins from DMSO treated or staurosporin treated soluble proteins. And on the y-axis you have here is the -10 p-value of the different replicates and the difference between the two groups. And in blue, I've highlighted all the kinases, and as you can see, a lot of these are kinases, and a lot of these are kinases which are known to be targets of staurosporin. So this kind of serves as a way to validate the method, that the method works, and you're seeing some of the targets that you expect. So I want to scale this up and basically cover a large portion of the proteome, and one strategy, and the strategy that I'm utilizing, is actually taking organs from rats and extracting their soluble and active proteins. So actually my collaborator is the one who's sacrificing the rats and extracting these organs. And this serves as a way to actually obtain large amount of material for these assays, but also to potentially cover a large amount of the proteome. And so I'll demonstrate some of my results from initial experiments by using this strategy. And the way I initially checked to see that these lysate extracts were active and that we're able to screen for drug target engagement is by basically testing against the known compound staurosporin. And the way I perform this assay is I aliquot the lysates into 9-6 well plates, which are then treated with either a control or different drugs. And then the plate is heated at different temperature blocks, afterwards which the different temperature blocks are pooled into one. As I mentioned earlier, this integration approach, and then from which I remove the insoluble proteins, and then I perform my in-plate sample prep cleanup using recent magnetic beads, onto which the soluble proteins are aggregated. And so this serves as a way to clean up the proteins. And I work with recent bioscience to actually use these hydroxyl beads because it works really well in 9-6 well format. And they're a good size. They're not too small, so they are very easy to separate via magnet, in my experience, compared to some other beads. And and then once the proteins have aggregated on these beads, I perform an overnight trypsin lysine digest to generate tryptic peptides. And then another advantage of this workflow is that I use EVO1 or EVOCEP nano HPLC system coupled to Exploris mass spectrometer. And this is a high throughput nano LC system. So I take the digest from these plates and take the digest and directly put them onto EVO tips. And these EVO tips, which are kind of like stage tips, they work with the EVOCEP so that the cleanup is kind of bypassed because the samples are loaded directly onto these tips for analysis by EvoSep and the mass spectrometer. And just screening these organs, I was able to actually cover over 8,000 protein groups. So just to give you an idea of the coverage I had. So I'll just give you some results from, for example, the muscle tissue extract. I see that, when I perform this experiment, we can see that we see a lot of the kinases which are expected to be affected by staurosporin, including a lot of these PRK subunits. And I've highlighted all the kinases in blue. But I think what is also most important is we see some of the off targets or some of the known off targets of staurosporin, including this furochelatase. And this is actually a common off target of a lot of clinical kinase inhibitors. And it's actually been shown that this off-target effect is known for the side effect of phototoxicity. A lot of cancer patients receiving some of these kinase inhibitors get. So this was a nice way to kind of validate the method in these extracts and to check that the extracts were working actually. Of course, I performed these in various different extracts and getting a lot of engagement of different proteins and a lot of these is expected kinases, but also non-kinase proteins, which is of course interesting because it's good to know, what could be potential off targets as well. Additionally, it was also interesting to see that the liver enzymes are highly active as well. And this was measured by basically by looking at the number of off targets or statistically regulated proteins between control treated and staurosporin treated liver lysates. And this was really dependent on the concentration of the inhibitor that you put into this lysate. But more importantly, this was also actually limited by the time. For example, even after a 30-minute incubation of the lysates with staurosporin, you can lose a lot of these inhibitors, indicating high enzymatic activity within the livers. And this is not so surprising because toxicology studies in rats and mice have shown with intravenous staurosporin injection that the drug actually breaks down within an hour of injection into 5 different unknown metabolites. So this actually served as a nice way to check that the liver enzymes are highly active. And that's good because the liver is actually a very good organ to potentially determine protein targets from. So with that, I will just end the talk. I just want to wrap up by saying actually that these organ extracts, protein extracts, are a good source to perform some of these type of protein target engagement experiments. And it's really facilitated by this thermal proteome profiling based method, which can be scaled to a 96-well format. And this works in line with doing a 96-well sample prep based on protein aggregation and magnetic beads. And of course, this is also facilitated by using these Evo tips on the EvoSep1 mass spectrometer, which is also done in a 96-well format. So it all comes together to kind of do really make a very robust workflow. And currently I'm testing different classes of drugs on these different lysates. And of course, one can utilize additional organs as well, if you can, of course, extract them in a nice and clean manner. And with that, I want to thank Jesper Olsen for giving me the opportunity to perform this research. So it's been really helpful, as well as members of the Olsen group who've all been really helpful in providing feedback and helping develop some of the research. And also want to thank my collaborator from the Department of Neuroscience here at Copenhagen University. She's really good at extracting the organs and really good at extracting them in a clean manner. And of course, I additionally want to thank Estoin and Justin for sponsoring this session, but also working with us to try some of these different magnetic beads and providing some good solutions for these 96-well based assays. And with that, I want to thank you for your time and look forward to any questions you might have. Thank you.